Welcome to Lecture 9. In this lecture, we are going to be talking about biotechnology, specifically how biotechnologies are going to be able to help extend the human life expectancy. So the course so far, we have been talking about how human longevity was primarily limited by things like food sources, death caused by bacterial and viral infections. However, because of the use of agriculture, discovery of antibiotics and vaccines, those have significantly increased the human longevity. And in the past few lectures, we talk about how organ transplant further increased the life expectancy of people who suffer from organ failures. But the last frontiers that are kind of still waiting to be conquered is cancer and other terminal illnesses, many of which still don't have cures yet. And this is where biotechnology is going to come in. We are at the cusp of using biotechnology to potentially treat many of these diseases. And if we are able to find cures for cancers and for terminal illnesses, then human life expectancy is going to be increased significantly. So what is biotechnology? Biotechnology is exactly what it sounds like. It's the technology based on biology. Okay, and it's an attempt to control, to alter, and to harness cellular and biomolecular processes to develop technologies as well as products that could help improve human lives and, in some cases, the health of our planet. Biotechnologies can be used as therapeutics. We can use biotech to help heal the world by harnessing nature's own toolbox and using our own genetic makeup to heal and guide lines of research by, for example, reducing rates of infectious disease, save millions of children's lives, as well as changing the odds of serious life-threatening conditions that are affecting millions around the world. We can also use biotechnology to tailor treatments to individuals to minimize health risks and side effects. We can create more precise tools for disease detections and, in some cases, combat serious illness and everyday threats confronting the developing world. Streamlining biotechnology can also be used to streamline steps in chemical manufacturing process by more than 80%. And we can lower the temperature for cleaning clothes, for example, and potentially saving $4.1 billion annually. We can also use it to improve the manufacturing process efficiency to save 50% or more on operation cost. Other use include reducing the use of and reliance on petrochemicals, and instead we can switch to using biofuels to cut greenhouse gas emission by 52% or more. We could decrease water usage and waste generation, as well as tapping into the full potential of traditional biomass waste products. Biotech can also be used to feed the world. We can engineer crops to create higher yields with fewer inputs, lower the volumes of agricultural chemicals required by crop limiting the runoff of these products into the environment, as well as to use biotech crops that would need fewer applications of pesticides, and that will allow farmers to reduce tilling farmlands. It's also possible to develop crops with enhanced nutritional profiles, and that would help solve various deficiencies, such as vitamin A deficiencies and nutrient deficiencies, and we could produce food free from allergens and toxins, such as mycotoxins, and improving food and crop oil content to help improve cardiovascular health. So let's take a look at golden rice as an example. Vitamin A deficiency is common in poorer countries. And when you don't have enough vitamin A, it could cause people to have nighttime blindness, okay? Because vitamin A is important for your eyes to function properly, as particularly in low light settings, okay? So vitamin A deficiency is the leading cause of preventable childhood blindness in these poorer countries. And what happens is combine that with low vaccination rate that could spark an endemic for infectious diseases. So in some countries, each year is estimated to kill 670,000 children under the age of 5 and cause an additional 500,000 cases of irreversible childhood blindness. And you can see here from this world map that in developing countries, that's where you see very severe prevalence of the vitamin A deficiency that we were talking about, and there was no good fix for this problem. That is until the creation of what's called golden rice. Golden rice is basically a genetically engineered rice that is created to make beta carotene, which is something that can be used by the body as a building block for vitamin A. So there were some controversies with the development of golden rice. For example, an early issue that was associated with golden rice was that they didn't have sufficient vitamin A in it. Okay, so later on they came out with a newer version called the golden rice number 2, which has 23 times more beta carotene than the original. So this was supposed to solve the vitamin A deficiency problems, right? Because now you can grow these rice in regions that have low vitamin A deficiency, and if people consume the rice, then they would have sufficient vitamin A intake. But Greenpeace opposed the use of any patent genetically modified organism, in this case the genetically modified rice, in agriculture. And they opposed the cultivation of golden rice because that would open the door to more widespread use of GMOs, or genetically modified organisms. Some worry about corporate control of agriculture in poor countries, which is typically where the golden rice will be grown. And others argue that maybe it's sufficient to just get vitamin A from other food sources. But that's not always easy because those foods are typically only available seasonally, and often they're too expensive for poor families in underdeveloped countries. So now let's take a look at genetic diseases, which is the goal of how biotech can be used to treat these disorders. A genetic disorder is basically a disease that is caused by abnormalities in a person's DNA, and cancer certainly falls within this category, but it's a little bit more complicated with cancers because there are usually multiple abnormalities in the person's DNA. So we are not quite there yet to use biotech to cure cancer. But for other diseases, it's only a single mutation in just one gene, and that is something that would be treatable potentially with biotech. And in some cases, there are an addition of an entire chromosomes, or there are some chromosomes that are missing, all of which can create genetic diseases. So take something called sickle cell anemia as an example. Over here, this is what your normal red blood cells look like, and red blood cells are responsible for carrying oxygen in the body. And what happens is if you have a single mutation in one of your genes, then it will cause your red cells to look like that. Or the sickle cell shaped, and in that shape, the red cells cannot carry oxygen, and the person becomes anemic, okay? Meaning they are going to experience shortness of breath, they're going to feel tired because they don't get enough oxygen being distributed throughout the body, and for something like that, where there's a single a mutation that's causing the disease, that makes it a prime target for using biotech as a treatment option. And we're going to talk about how that's possible later on. Here is another example. There is something called beta thalassemia. In this case, there is again a DNA mutation that causes your hemoglobin to be missing two parts. Okay, so hemoglobin is shown here, and these are the proteins that are inside your red cells that is responsible for delivering oxygen in the body. And in this mutation, it causes two of the chains to be missing. Okay, so here you can see that's the mutated version of the hemoglobin. And people with this beta thalassemia, they are severely anemic, okay? And they actually require constant blood transfusion in order to stay, relatively normal, okay? Without transfusion, they would not be able to carry on with their normal daily activities. And with blood transfusion, it's even though it's a solution, but it's not a permanent fix, because if you keep on getting blood transfusion all the time, then you actually become iron overload, and that requires other treatment as well. So something like this, again, hopeful candidate for biotech as a treatment. Huntington is another disease. You might have heard of that. It's a brain disorder that affects the person's ability to think, talk, and move. And it's actually caused by a mutation in a gene that is on one of your chromosomes, chromosome 4. And that causes an abnormal accumulation, a buildup of protein clumps in the brain cells, and that will cause it to die over time. The thing with Huntington is that the symptoms of Huntington don't usually show up until the person is after 40 years of age, and by then most people might already have a kid or two already. So they already might have passed on the bad gene onto the next generation by the time they realize they might have Huntington. Another example is hemophilia. People with hemophilia, they are missing blood clotting proteins. Basically meaning if they have a cut, they won't be able to clot the blood easily, and that could cause them to lose a lot of blood, and sometimes it's life-threatening if the bleeding is happening internally, and so they need to have injection of the clotting proteins in order to stop the bleeding. And again, this is caused by genetic mutation in a gene. So just like the other cases, it could potentially be fixed by biotech. And another example that we talked about before, actually, the SCID, which stands for Severely Combined Immunodeficiency. You might remember the bubble boy that we've talked about in lecture 2, I believe. These are people who are born without immune system, and that actually is a mutation found on the X chromosome, and so there has been some cases where people are using gene therapy to fix this problem. Okay, so the key thing to take away here is if a disease or disorder has many different mutations, such as cancer, for example, then it's not going to be easily fixable with gene therapy, at least not at this stage, okay? But if you have a genetic disorder that involves a mutation in a single gene, okay, or in some cases in a single spot, then that makes it a prime target for gene therapy, for biotech, to potentially be a treatment. So let's go through some basic concepts related to heredity. Now, we know what a gene is already. Okay, a gene is basically a section on a chromosome that contains instruction to create a particular trait. Okay, so if you need a refresher of what a gene is, you can go back to lecture 3, where we talked about evolution. In that lecture, we discussed what a gene is. Now, what I did not tell you at the time is a gene can have multiple versions. Okay, for example, you can have a gene for eye color, and there would be multiple versions of that genes that cause people to have blue eyes, brown eyes, green eyes, and all the other colors. So a different variant, a different version of a gene is what we call an allele. Okay, so there might be several different allele that exist in the population, but each person can only hold a maximum of 2 alleles. Okay, because we inherit half from each parent, so there are only enough space for 2 alleles of a gene at the same time. So let's say we have an allele that codes for brown eyes, and that's represented by the letter capital B. And then we have an allele for blue eyes, and that is represented by the small b. Okay, so someone who is big B, okay, like here, that's gonna be someone with brown eyes. And if another person has small b, then the person would have blue eyes. So you might be wondering, what about someone who inherit a big B from one parent and a small b from another parent? What eye color would that person have? Well, that has to do with the concept of dominance versus recessive. Some alleles are dominant alleles. That means it will override the recessive copy when both are present. So if you have big B and small b, the big B is the dominant version, and then it will override the small b. So in our example, big B represent brown eyes and small b represent blue eyes. So if you have both, the person would turn out to be brown eyes. Okay, because again, the big B will override the small b. That means if a person has blue eyes, then you would know they are carrying both small b in their gene. However, if a person has brown eyes, then you actually don't know whether they are big B or big B small b. Okay, so you would have to look at their family history to kind of find out. So let's take a look at what's going on here. Let's look at this example first, okay? So let's say there are 2 people. One of them have brown eyes, and I'm going to tell you that the person is big B. And then the other person, the spouse, is going to be small b, which is blue eyes. Okay, so let's say this is the man, and let's say big B is the man, and small b is the woman, and they're going to have a kid. Okay, so what happens is the man is going to be passing on only the big B, okay? Whereas the woman is only going to be passing on the small b. So the sperm from the man will be carrying the big B, and the egg of the woman will be carrying the small b. And when the sperm and the egg come together to create a baby, then the baby is going to be big B, small b, like that, right? Half of it inherited from the father, in this case the big B allele, and half of it inherited from the mother, in this case the small b. So the child is going to have brown eyes, okay? Because the dominant allele here is the big B. Now let's take a look at another couple, okay? They are both brown eyes, big B, small b, and big B, small b. Okay, so when the man makes the sperm, half of the sperm will be carrying the big B, and the other half will be carrying the small b. And same thing for the woman, half of the eggs will be carrying the big B, and half of it will be carrying the small b. So if this sperm fertilizes that egg, then the result will be big B. If this sperm fertilizes this egg, then the result will be big B, small b. And then if this sperm and this egg comes together, it will be big B, small b again. And finally, if this sperm and this egg comes together, it will be small b. So statistically speaking, there is going to be a 25% chance, okay, 1 out of 4, right? 25% chance that the baby is going to be big B, okay? And then there's going to be a 50% chance, okay? 50% chance that the baby is going to be big B, small b, and then there's going to be a 25% chance again where the baby is going to be small b, small b. So if you put these together, there's a 75% chance that the baby is going to have brown eyes. Okay, I'm going to erase this. It's going to have brown eyes, 75%, okay, chance. And then there is going to be a 25% chance that the baby is going to have blue eyes. Okay, so the key takeaway here is that a person is only going to be able to pass on one of the two genes that they are carrying. Okay, and you're going to be able to determine half of the genetic material of a child, and then the other half it's going to be from the other parent. So mutation that causes disease can either be a dominant mutation or a recessive mutation. If it's a recessive mutation, then it takes 2 copies of the faulty gene in order for the disease to manifest. Just like the small b, the blue eyes, right? It takes 2 copies of the blue eye gene for the blue eye to show up. Okay, so if it's a recessive disorder and you only have 1 copy of the faulty gene, then you don't have to worry about it because the healthy version of the gene, the normal version, will override it and the disease will not show up. So you will be a carrier of the disease, but you won't actually have the disease. On the other hand, if the genetic disorder is as a result of a dominant mutation, then only 1 copy of the faulty gene is enough for the disease to manifest. So for that reason, dominant disorders are usually more prevalent in family compared to recessive disorder. Hey, again, dominant disorder, 1 hit is enough to make the disease happen, but for recessive disorder, you will need both copies of the faulty genes. Let's use sickle cell anemia as an example again. Okay? Sickle cell anemia is where you have a single mutation that causes your red cells to go from this shape to the moon shape, okay, making you anemic. Okay, so that is an autosomal recessive disorder. So autosomal means is carried on the chromosome that is not your sex chromosome, so not X or Y. It's carried on one of the chromosomes from 1 to 22, and it's a recessive disorder, meaning you will need 2 copies of the faulty genes before the disease is going to show up. So here we have a mother who is a carrier, which means she has 1 copy of the faulty gene. You can see over here, right? So we will say she is a big S and small s. Okay, so big S we will say is normal version of the gene, and small s is the sickle cell. Okay, sickle cell gene. Okay, so she is a carrier, meaning she has 1 big S and 1 small s. Same thing for the dad. The dad is also a carrier, so big S and small s. Okay. So similar to the brown eye and blue eye situation, the mother, half of her eggs are going to be carrying the big S gene and half of it is going to be carrying the small s gene. Whereas the father, same thing, half the sperm is big S, half of it is small s. And if you do the matching, you will see you can get big S, big S, big S, small s, big S, small s, or small s, small s. And so 25% chance the child will be big S, big S. So this is big S, big S, meaning both copies of the genes does not have the mutation. Okay, the mutation is shown here with the red markings. Okay, so both copies of the genes are normal, and the person is going to be normal. These 2 kids, they are going to be big S, right? The normal copy on the left, and small s, which is the faulty copy. And they are also going to be okay because this is a recessive disorder, and that means you, if you only have 1 copy of it, in such as this case, the normal copy will override it and disease will not show up. And for the last one, then is small s, and this is the person who will have the sickle cell anemia. So that's kind of interesting because even though both parents are okay, both parents do not show any symptoms of the disease, they are still able to have a child that has the disease, okay? And that's because both of them are carriers. So mathematically speaking, there's a 25% chance that their child is going to be sickle cell anemic. And that's what genetic counseling is, okay? If there are certain genetic condition that runs in the family, people might consult a geneticist and they will look at your chromosomes, basically your DNA, and they will tell you what's the probability of your child having a particular disease. And if the probability is high in some cases, then the couple might decide not to have the kid. Huntington is another example, and this one is an autosomal dominant disorder. So again, autosomal just means it's not transmitted through the sex chromosomes, and dominant means it only takes one copy of the faulty gene for the disease to show up. So here we will use big D as the disease, the Huntington disease, and small d is the normal one. Okay, and I use capital D for disease because it's a dominant disorder, right? So if you look at the mom here, she is normal, so she's going to be small d. And in the father, the dad, he has Huntington disease. So in the genetic, we can see there is the faulty gene right there. So there's going to be a big D and a small d. And if you do the matching grid, okay, it's called a Punnett square. You can see the mother's eggs are all going to be small d or small d. And then for the dad, half of it is big D, half of it is small d. And if you do the matching, you can see there's a 50% chance, a 50% chance for the child to carry the big D, and that's what these are. Okay, so they will have Huntington disease, and here the other 50% is basically the normal individuals. And as I mentioned earlier, the problem with Huntington disease is by the time the father manifests the symptoms, okay, he's probably going to be in his mid or late 40s, and by then he would have had kids already, and so the dilemma here is, should the kids get tested? Okay, and if the kids are young, like in their early 20s, for example, and they tested that they will eventually have Huntington, right? That raises a lot of ethical questions, right? Things like genetic discrimination. Would insurance company deny to cover them, not knowing that they might develop Huntington later on, right? Would employers not hire them, knowing that they might not be able to do their jobs later on in life, right? So that sort of questions certainly comes to mind when we talk about genetic counseling, that kind of stuff. So the first biotechnology we're going to be looking at is cloning. So let's take a look at something called somatic cell nuclear transfer. Somatic just means a cell that is not sperm or egg, and in this diagram you can see we have a donor. That's the sheep where we'll be taking the nucleus from. So the nucleus is where the DNA is going to be, and so you will take the nucleus from the somatic cell of this sheep, and then you will put it into the egg of another sheep. So after taking the egg from this sheep, you will remove the original nucleus, and then you will be injecting the nucleus from the donor cell, and that creates an embryo carrying the same genetic information as the somatic cell donor. And then you'll be implanting this into a surrogate mother, the surrogate sheep in this case, and that will give birth to a clone sheep that will be genetically identical to the sheep that was donating the nucleus. Okay, this other sheep has nothing to do with the clone, okay, genetically speaking. Okay, it's only provide like an empty eggshell, so to speak, for the nucleus to be in it. So that's how the first cloned mammal was created. The name was Dolly, and it was the first successfully cloned mammal using an adult somatic cell. It was born after 277 eggs were attempted, and it created only 29 viable eggs, and only 3 of these embryos survived until birth, and then only one survived to adulthood, and then that's Dolly right there. Now, the normal life expectancy of this type of sheep is around 11 to 12 years long, but for Dolly, the clone sheep, she only lived to 6.5 years, and that's because when you take the nucleus from the donor cell, okay, so remember this was the donor cell, okay, the age of the cell does not get reset when you transfer it to an egg. And so when Dolly was born, even though the birth age is 0 years old, but the cellular age is the same as that of the donor sheep, and that's why Dolly only lived for 6.5 years. So when it comes to using biotechnology as therapeutics, we're not talking about reproductive cloning. Okay, we're not going to be running around cloning humans. Not only is that obviously an ethical problem, but technologically speaking, we're not even close to doing that. What we are talking about is something called DNA cloning. Okay, so DNA cloning is a molecular biology technique that makes many copies of a piece of DNA. Okay, so for example, if we want to make a copy of a gene, we have the technology to do that. Copying DNA, as you know, is a normal cell process. You do that every time the cell splits. You remember in lecture 3, I think, we talked about how a cell is going to split into 2 other cells, but before it does that, it's going to need to copy all of its genetic material. And basically, scientists are exploiting that process, and we use that to basically clone the gene of interest. We take advantage of the fact that DNAs are complementary, meaning if you know one side of the DNA strand, you can predict what the other one is. And that allows us to copy basically any parts of a DNA as many times as we want. So remember, bacteria, they have plasmids, right? So these circular DNA are part of the bacteria's genetic material, and you know that the bacteria like to exchange the plasmids regularly, right, through conjugation. And usually these plasmids are going to give the bacteria some kind of survival advantage. That's how superbugs are created, right? So that's something that we talked about. Now, what we want to do now is to create our own plasmid, okay? So here you can see a generic plasmid, and there is a section in the plasmid that we can insert any genes we want into it. Now, the exact science is kind of complicated, so we're not going to go into it in this course. But once we created a kind of custom-made plasmid, we call this recombinant DNA, okay, recombinant DNA. Then what you can do is you can feed the plasmid to the bacteria, and then the bacteria is going to copy that every time it divides. So how can we use that in something like gene therapy? Let's go through the basic concept of gene therapy. Gene therapy involves introducing genetic materials into the patient's cell. Okay, so there are 2 kinds of gene therapy. We have gene therapies that involve somatic cells. So these are somatic gene therapy, which means you are going to be transferring a section of DNA to cells that does not involve sperm and eggs. Okay, these kind of gene therapy will only affect the patient, but not future generation. Now, germline gene therapy is a lot more controversial because now you are modifying the sperm and the egg of the person, and so this effect is going to be passed on to the patient's future generations. So the goal of gene therapy is to do one of the following. Okay, you can replace a non-functional gene. Okay, you can suppress the activity of a gene that is overly active, and you can help modify the clinical course of a certain condition. So let's take a closer look at each one of these things. First one, replacing a non-functional gene is probably the most straightforward in terms of understanding how it works. So if there is a mutation, if there's a mutation that results in a loss of function in a protein, okay, so remember, genes are instruction to make proteins, and if there is a mutation in a gene that causes the protein to not work, and that results in some kind of disease, then what happens is we are going to be able to replace the defective gene with a functional gene. Okay, so here is a cell with a non-functioning gene, and we can clone the gene of interest using the plasmid, the bacteria, and then we can deliver that gene into the person, and then the person will be able to make the functional protein once again, and in theory that should be able to fix the problem. These conditions are usually recessive, and the reason for that is because if the disease is dominant, then you will have to replace both copies of the defective gene in order for it to be successful. So more on that. This kind of gene therapy is only successful if the effects of the disease are reversible or have not resulted in lasting damage to the body. Okay, so things like sickle cell anemia, beta thalassemia, and hemophilia, as soon as you reintroduce the functional gene, that should help reverse the symptoms. But if we're talking about some disease that already have caused, say, brain damage, then gene therapy will not help to reverse the brain damage because it's already done, right? Another another goal is to suppress a gene. So these are mutations that causes the gene to be overly active. We talk about this in the cancer unit lecture, where if you have a gain of function mutation, that could cause a cell to divide uncontrollably, and that's basically what an oncogene is, right? Resulting in cancer growth. So in this case, here we have an overly active gene, and it's possible to insert a blocking gene and deliver it into the cell. And once the blocking gene is inserted, it will suppress the overly active gene, and that will hopefully restore normal cellular activity. So something like this might work in an early stage of a cancer where it has not spread yet, and it can be a means to control the growth of the tumor. And finally, it's possible to insert a DNA, a sequence, a gene, into a diseased cell that caused the cell to die. And again, this would be something that would be suitable for cancer. Okay, you can insert a so-called suicide gene, and once it enters the cell, it's going to trigger cell death. Okay, and that would be good for killing off cancer cells. It's also possible to insert a gene that cause the cell to attract the white cells to attack them, and that way you can kind of harness the energy of your own immune system to attack the cancer cell. So this is what it looks like. You have a disease cell right here. You're going to insert a suicide gene. As soon as the gene goes into the cell, it triggers programmed cell death, and then the cell would die. On the other hand, you are going to introduce a marker gene that causes the cell to display all these markers on the surface, and those markers will attract the white cells to come along and destroy it. So we can do gene therapy through what's called in vivo or ex vivo. So in ex vivo, what you do is you would take the cell from the patient's body. Okay, for example, you can take cells from the bone marrow of the person, and you will genetically modify those cells in the lab, okay, in a petri dish, and then after the cells have successfully take up the gene, you would re-implant them back into the patient. So this is called ex vivo, which means outside of the body. And in vivo, you would use a vector. A vector is something that is responsible for delivering the gene into the cells in the body, usually some kind of virus, and the DNA will transfer to the target cell, and everything is happening within the patient's body. So in vivo. Now the challenge of gene therapies are as follow. Okay, target delivery, immune response, interaction with other genes, and cost. So let's take a look at each one of these. It is crucial for the new gene to reach the intended cells. Okay, so once you put the gene into the body, you have to make sure that it is inserting it in the right cells and in the right place. If you insert the gene to the wrong cell, then not only is it going to be an ineffective, inefficient treatment, but it could also potentially cause other health problems. And even if you insert the gene into the right cell, you have to make sure that the gene is turned on, it's activated. Otherwise, you put the gene in and it's not switched on, then it's not going to be able to exert its effect, right? And sometimes because these kind of genetic materials are kind of foreign, so cells they have natural mechanism to kind of obstruct the process of turning on foreign genes, and so you have to find a way to circumvent that. Like I said, usually we use a virus as a vector to deliver the gene. So the gene is going to be fed to the virus, and then the virus will infect your cells. Okay, so this of course is going to be a virus that is just an empty shell. It doesn't cause any disease, and then the virus will insert the gene into the nucleus of the target cell. Now, because we are using a virus in some cases to deliver the gene, so the immune system is going to be triggered. Okay, and in some cases, the immune response is quite drastic, and it could cause some harm in the patient. And the challenge here is to find a way to deliver the gene without triggering the immune system. When you introduce a new gene into the body, again, we need to make sure that it's going to be inserting in the right place. So, for example, let's say I have a like a regular gene here, okay, and then I want to insert this blue gene, okay, but if we're not careful, then the blue gene might accidentally insert itself into the middle of another gene. So you might be able to turn on the blue gene, but now you disrupt the function of another gene, and that could be a problem. If that red gene here is important for regulating cell division, then the therapy could result in abnormalities related to cell division, which could potentially result in cancer. So you have to make sure that the gene you're delivering is going to be inserted into the right place. And just to overcome all those three challenges is going to require a lot of resources, and it's actually going to cost quite a lot. And in many cases, the gene therapy are for disorders that are kind of rare, and these requires tailored approach in many cases. And you can imagine the cost is going to be very, very high. And you can click on this link to see some of the most expensive trials that are going on. Just to give you an idea, depending on the type of gene therapy we're talking about, it ranges from about $500,000 per treatment to as much as $1.5 million. And some therapy actually requires ongoing cost. For example, therapy used for a type of spinal muscular atrophy cost about $750,000 in the first year, followed by an annual cost of $375,000, and that is for life. And so even though these are treatments that are potentially life-altering, it's not necessarily accessible and affordable to most people. Now, some company they have adopted what's called a pay only if it works approach. That is, if the therapy doesn't work, then you don't have to pay the money. The drug company will absorb the cost. But what's considered as works is a tricky definition. So that's it for biotechnology. Thank you for watching.